expertise in it. To introduce Eric, or Dr. Van Nostrand, he is assistant professor and CLIP scholar in the Verna and Mars Department of Biochemistry and Molecular Biology at the Baylor College of Medicine, which is in Houston, Texas. His lab studies how RNA-binding proteins regulate RNA processing. And Dr. Van Nostrand did most of his undergrad studies in biology at MIT and received his PhD in genetics from Stanford and was a postdoc and Merck Fellow and Damon Ryan Scholar as well at the University of San Diego, California, San Diego. So very excited for Eric's speech, and Eric, please take it away.
Yep. Oh, thank you very much for the introduction and the invitation to come talk to you all today. Let's do that. Is that working? If no one yells, I'll assume that that's working. Okay, great. Yeah, so thank you all for coming. And I'm excited to tell you guys about some of the work that we've been working on over the past few years, trying to build what we call a large-scale view of RNA biology. Before I start, so as disclosure, I'm co-founder of a company that's building some kits and service products in the RNA genomics space. And I'm happy to answer questions about that afterwards if anyone has questions, but just disclosure, all that I'm going to talk about today is our academic work, primarily at UCSD. So what we really focus on is sort of what happens after an RNA is transcribed from the DNA, but before you have the ultimate protein function, right? And I'll talk more about what I mean in terms of RNA processing and what those steps are on the next couple of slides, but just to emphasize how important these steps are, there's now a huge laundry list of human diseases, ranging from different types of cancers to things like neurodegenerative diseases and ALS, muscular dystrophy, and many others that we now know are caused or can be caused by either mutations in RNA processing regulators or RNA regulatory elements. Right? And so I think it's becoming clear that RNA processing is sort of critically important for a whole host of human tissues and cell types and physiology in general. Oops. So RNA processing actually means a lot of different things, right? So after an RNA is transcribed in the nucleus, it undergoes things that can change the sequence of the RNA, so things like splicing, where your introns are removed and exons are joined together. So that can change the sequence of the protein that's made. You have steps that regulate where the RNA is in the cell. So that's things like regulation of export from the nucleus or localization in the cytoplasm. You have regulation of how long that RNA is stable for in the cytoplasm. So regulation of the half-life, targeted degradation, things like that. And then you have regulation of translation itself, of initiation and elongation of translation. And what I'm going to talk about today, the key thing is that each of those steps in the picture here is controlled by all these different colored ovals and circles, right? So these are RNA binding proteins that bind to RNAs, some recognizing sequence, some recognizing secondary structure, or a mix of both. But they regulate RNA processing in different cell type specific or disease specific or even condition specific ways. And there's been a lot of work over the past few years trying to understand and characterize how many RNA binding proteins there are. Computational efforts, as well as experiments where people just pull down RNA and mass spec what's associated with it, have identified that there's over 1,500 RNA binding proteins in human. And as I said, we now know that mutation in lots of these RNA binding proteins actually causes disease. And so it's becoming very interesting and important for us to understand what these RNA binding proteins are doing and how they're regulating their targets. And as one sort of final background to give a sense of the way that we think of the potential for understanding these targets, the example that everyone likes to use now is spinal muscular atrophy, or SMA. And so SMA is a really terrible disease. As motor neurons die, this leads to muscle wasting, ultimately leads often to fatality because the muscles in the lungs stop working properly. And through a lot of work led by Adrian Krainer's lab, along with many others, they've characterized this disease, that the molecular cause of the disease, and I'm oversimplifying this for this short talk, but there's RNA binding proteins like HNR and PA1, and they bind to RNA elements in a gene called SMN2. And when they bind to those elements, they cause this exon in red here to be excluded 90% of the time. So in normal in normal tissue, this exon is excluded. It turns out that if you block those RNA binding protein interactions, in this case here showing using an antisense oligonucleotide, this exon is now included 90% of the time, and that actually treats SMA. So the ASO version of this is called Spinraza. It was a drug that is actually kind of famous now because it's one of these $100,000 per dose drugs, but it made it through clinical trials a few years ago and is now a multi-billion-dollar drug. And so when I talk about, for the rest of the talk, I'm going to talk more about sort of a mechanistic perspective and some technologies. But big picture, this is sort of the way that we think about this in the future, that if you know enough about the biology of the system and what the RNA interactions are, these RNA binding protein interactions, even individual ones, can actually represent therapeutic targets if we know enough about them. And so what I'm going to talk about today is basically how my postdoctoral work, and then a little bit about how my lab is trying to approach this question of, it's pretty common now that we have an RNA binding protein that we're interested in. We know that it causes some disease, or if you knock it down in cell culture or knock it out in a mouse, it has some sort of phenotype. And the question is, how do we fill in those gaps, right? And the way that we've approached this is to try to split it up into sort of two separate questions, right? Of how do we figure out, well, what are its targets? What is the regulatory network that it's controlling? And then as a second question, what is the subset of those maybe thousands of targets that are actually the ones that are the most important for the phenotype? And so that last question, I would say, is still a very difficult and open question. What I'm talking about today is more our efforts to try to do a better job at the first step. So developing methods to do a better job of understanding what these targets are, and then our efforts to sort of build this large-scale RNA regulatory map, which sort of sits in between of trying to understand globally what is an RNA binding protein doing. So broadly, what these methods are doing is, we have an RNA binding protein that we're interested in. The cell is lots of RNAs decorated by lots of different RNA binding proteins. And so what we're trying to do is we're trying to isolate the RNA binding protein of interest along with sort of the small region of RNA that's being bound by it. And so I'm not going to go too much into the history here, but to give you a very broad sense of these methods, they're called Crosslinking and Immuno Precipitation, or CLIP. We developed an improved method, which we called enhanced CLIP, or eCLIP. And I'll just very quickly go over the high level steps to give you a sense of what the kind of data is that I'll show. So these CLIP methods start with a UV cross-linking step, so that covalently links the protein to the RNAs that it's bound to. Then there's a bunch of steps that are basically enrichment steps. So we fragment the RNA to get rid of the rest of the RNA and leave just the RNA that's bound by the protein of interest. We take an antibody against the protein and immunoprecipitate it. So we're trying to enrich for that specific protein. CLIP actually includes denaturing protein gel and a transfer to a nitrocellulose membrane, like a Western blot. The idea there is that it helps to remove non-cross-linked RNA, as well as to get rid of other proteins that may have come down in the immunoprecipitation that are of a different size and things like that. The rest of the protocol is essentially like RNA-seq. It's a library preparation. You have single-strand RNA at the beginning here, and what we need to do is put adapters on it, reverse transcribe it, PCR amplify it, and get enough material to sequence. After we sequence it, then there's a computational challenge, right? We need to say, well, what are the regions that have more reads than you expect by chance? And so we have computational pipelines to try to identify what do we mean by chance, right? And so we try to find regions that, like here, that have more reads than neighboring regions, that have more reads than paired controls, like input or IgG controls. We try to build different methods to identify those significantly enriched peaks. And I'll talk about those a little bit more in a second. To give you a sense of what the challenge is here, and for time, I'm not going through the history of CLIP methods here, but the challenge is basically that the amount of RNA that you're getting is on the order of picograms to maybe a couple nanograms. So that in many cases, for many RBPs, is almost like single cell levels of RNA-seq. So it's actually a relatively difficult library preparation question. And so really the challenges and improvements that people have been making recently in CLIP really have to do with these library preparation steps and making it better, improving our ability to get library out of the very small amount of RNA that's actually being pulled down. If you're interested in more details, obviously you can reach out to me, and I'm happy to point you to the right protocols and stuff like that. But there's a really great review written by Janusz Ule, who did much of the early development of CLIP methods and really interesting studies with those. But they have a really great review going through the history of CLIP, published a couple of years ago now. Okay, so a couple of quick things, and this was a paper that we published a few years ago now in Nature Methods describing the method. So we showed that this worked, so we could do CLIP, this for RBFOX2, a splicing factor. We could then go to that next step and design antisense oligos. And if we treated cells with this antisense oligos, we actually blocked RBFOX interaction and we changed splicing of nearby exons. So this is actually sort of a complete pipeline that people can now do. You can take an RNA-binding protein, you can identify its targets, and then you can make these antisense oligos and actually block the RBP and see what the effect of that binding site is on splicing or different steps of different aspects of RNA processing. The other thing we showed was that one of the main challenges with CLIP had been a lack of proper normalization against inputs and controls and things like that. And I'll just very quickly show why that's important. So we're looking at CLIP data for SLBP, which is an RNA binding protein that plays roles in histone 3' processing. And what you can see here is that this SLBP binding site at a histone 3' is something like 2 to the 9th enriched in an IP versus a paired input control. But if you look at just an abundant RNA, so like a U12 spliceosomal RNA here on the bottom, you can see that there's lots of reads in the CLIP on the top, right? So if you just had that IP data set, you'd think that this has a ton of reads. It looks like a great peak. It's only when you have this comparison against a paired input control, then you can actually see, oh, it has a lot of reads, but it's really depleted versus this paired control. Now, this is not necessarily surprising to the biologists, because if you run an IP Western, it looks very pretty, right? You get a single band, but if you silver stain to stain for all protein that came down in that experiment, you see there's lots and lots of stuff that comes down in immunoprecipitation, right? IPs are enrichments, they're not protein purifications. But this has really significant effects for how you think about the data, because if you do just the standard analysis at the time that didn't include this input normalization, you got something like 30,000 clusters for SLBP. But when you incorporate the input, now there's only a couple hundred significantly enriched peaks, and these are all the ones that are at histone RNAs. So there's really a dramatic ability to improve the signal to noise by incorporating these paired controls and this paired input normalization. So I'm not going to talk really much about the rest of the method. Again, reach out to me if you're curious for more details or you have questions. But what I'm going to talk about for the rest of the talk is our efforts to then take this and really increase the scale and try to build these large-scale regulatory maps. So this was a large collaboration. So in Gene's lab at UCSD, we were doing eCLIP to try to map in vivo targets. Chris Burge's lab at MIT was looking at RNA-binding-seq to map in vitro RNA-binding motifs. Brent Gravely's lab at UConn was doing knockdown followed by RNA-seq to identify what gene expression and splicing changes occur when you knock down an RNA-binding protein. Shangdong Fu's lab at UCSD was doing ChIP-seq for a few RNA-binding proteins to start to explore the interaction between sort of their chromatin association and how that feeds forward into their RNA processing regulation. And then Eric Lucchier's lab in Montreal was taking the same antibodies that we use for CLIP and doing immunofluorescence. And so now you can see, where is this RNA-binding protein in the cell? What are its targets? And what changes when you knock it down, right? And so you can build this whole sort of landscape of what the RNA-binding protein's regulatory network is. So there's something like 1,200 data sets that we generated for 350 RNA-binding proteins. I show this just to emphasize that there's data available for splicing regulatory RBPs, translation regulators, stability regulators, RNA-binding proteins that came down in some of these, pull down RNA and mass spec the proteins that are on it. So they're RNA binding proteins, but nobody really knows what their function is or what they're doing in terms of RNA processing. So there's a wide array of data there. In particular for the CLIP, just to give you a sense, right? The idea was to make this as easy to use and as publicly available as possible. So this data has been available, for many years, even before our paper was, published. There's no embargoes or anything. You could take it and use it in your own studies for whatever you want. There's been a lot of uptake from the community already in making interesting use of these. But so for the CLIP, for example, there's 150 RNA binding proteins that have CLIP data in 1 or 2 cancer cell types. And if you go to the ENCODE project website, you can download the raw data, you can download the mapped BAM files, input normalized peak calls, even things like the IP Western images and other QC data, so you can judge for yourself whether you think the experiment worked well or not. And so it's been, it's been a really great resource, I think, for the community. We've gotten very good feedback. So what I'll talk about for the remainder is sort of some of the challenges and ways that we've sort of tried to think about, how do you analyze 150 RNA binding proteins at once, right? And what can we learn by comparing all of these different RNA binding proteins in parallel? So the first thing we looked at was just, where are their targets? And you can see there's sort of this wide array of binding from, RBPs where most of their targets are in introns to mostly in exons, coding sequences, 3' UTRs. In introns, you could be near the splice site or further away from the splice site. And at this level, it's not surprising that the ones that bind to introns tend to be things that are involved in regulating splicing, whereas things that bind to the coding sequence or UTRs tended to be more cytoplasmic regulators. But we're interested to push that a little bit further and look a little bit more fine-grained. And so we built what what are called metagene plots. So you're sort of averaging across all the transcripts in the genome and saying, well, where is sort of the typical peak for this RNA binding protein along this metagene plot? And so if you see here, you can see that when we line all these CLIP data sets up, there's very clear clusters where RNA binding proteins have very similar patterns of enrichment along these metagenes. And if we go in, we can sort of characterize what these clusters are and where they sort of have the largest spikes of enrichment. But if we zoom in here, for example, on the 5' UTR, there's really sort of two groups of RBPs there. So there's one in orange that includes things like the EIF proteins, FMR1, DDX3X. These are RNA binding proteins that regulate translation. And you can see that the peak of their enrichment tends to be right around the SAR codon, which matches their role in translation. The red cluster here, on the other hand, tends to peak more towards the 5' end of the transcript, so the transcription start site. And that tends to be RBPs like NCBP2, which is one of the nuclear cap binding proteins, so it binds the 5' cap on RNAs. CSTF2T, which binds involved in 3' end processing, but at a step when the RNA is circularized. And so the 3' end and the cap interact with each other. And so it's neat that this works because it means that if you clip an RNA binding protein that you don't really know very much about, just from where its targets are, from where its peaks are, you can actually get a pretty good guess as to what it might be doing mechanistically. We can also go in and then compare between data sets more explicitly. So just asking, well, for each data set, how many of its peaks overlap with other RNA binding proteins? And as you might expect, this pulls out things that are known complexes. So like U2AF1 and 2 is a complex that's involved in 3' splice site recognition. But we get complexes for the 5' splice site as well. Family members like TO1 and TO-like1 share a lot of their peaks. But then we get some interesting other clusters of RNA binding proteins that we didn't go in expecting that they would bind the same targets. This PCBP cluster here, there's an interesting story in the Nature paper on the chromatin side, actually. So Shengdong Fu's lab showed that there were some interesting overlaps between some of these RNA binding proteins at the chromatin level, and that sort of fed into their regulation of RNA processing. But HNRPL as well, there's another interesting cluster here that we're really just trying to start exploring now and trying to understand why do all these RNA binding proteins seem to share so much of their targets when, by function, they don't, at least superficially, seem to have sort of similar functions or similar roles. So again, it's a good way to sort of take a new RNA binding protein and try to have a sort of an educated guess as to what it might be doing. Finally, I'll tell a quick story about how you can integrate multiple types of data. So here I'm showing a single RNA, a single gene, and on the top we have knockdown RNA-seq of RBFOX2, a splicing regulator. And you can see here that this exon is included in the control, but is excluded when you knock down RBFOX2. And on the bottom, we have the CLIP data. So RBFOX binds to the intron near this exon and regulates its alternative splicing. So obviously, there's not just one exon like that, right? There's 138 exons in this data set that were excluded when you knock down RBFOX2. And so what we can do is we can take the CLIP tracks for all of those 138 exons and their flanking introns. And we can take all those CLIP tracks and then we can sort of average them. right? And we can say, well, here's sort of the typical profile or the enriched differential profile of RBFOX binding around exons that are excluded when you knock down RBFOX2, right? And these are what are called splicing maps in the field. And the idea is to sort of map out where is, say in this case, RBFOX2 binding. How does that binding at different places along the intron correlate with regulation of splicing? And you can see for RBFOX2 here, right, this is a common pattern that RBFOX binding to the downstream intron of an alternative exon tends to lead to inclusion of that exon. And so if you knock down RBFOX2, you get exclusion upon knockdown. We can then make that even simpler, right? If we remove the error bars there, which is an estimate of the variability, right? But if we remove those error bars, we can sort of summarize that as a single track to compare different RBPs. So we can do this, we have data now to do this for lots of different RNA binding proteins. You can go in, if you're interested in a single RNA binding protein, you can go look at this. Obviously there are other ones published in the literature as well. And so we now have, a good number of RNA binding proteins where we have these sort of maps available. You can see that they have different patterns. So the pattern of regulation of where RBFOX binds and regulates is different than PDB, which is more in the upstream intron, which is different from SRSF1, which tends to be more in the exons. And so, there's a lot of interesting individual RBP things that you can do here, but one question that we actually went and looked at was to go back and look at some of the general principles of splicing regulation. So there's been a model in the splicing field for a long time that very broadly you have HNRMPs and SR proteins as sort of competing with each other, that HNRMPs bind to these exonic splicing silencers and they suppress exon inclusion, whereas SR proteins bind to exonic splicing enhancers and they promote exon inclusion. And so we had enough of these HNRMP and SR proteins that we could actually compare and sort of globally see if this seemed to be true. And actually it generally was. So HNRMPs tended to be enriched at exons where if you knocked it down, the exons were included, so they're acting to suppress, whereas the SR proteins on the bottom here had the opposite effect, that they were more enriched at exons that were excluded upon knockdown. Now for me, it's interesting that this works, and as a general principle, it's true. It's also very interesting to see the ones that are sort of don't align with that pattern, right? So you can see here, for example, PCBP1 and 2 are HNRMPs, but their splicing map patterns don't really align with the other HNRMPs. They seem to either be sort of not really doing much at all or having sort of the opposite effect. And so we're trying to, it's interesting to then follow those up and try to understand, well, what's different about those two as opposed to the rest? Why are those two not showing the same pattern? So there's lots of other things in the paper integrating, the in vitro motifs and stuff like that I haven't talked about for time. But feel free to reach out if you have any other questions about that kind of stuff or want to chat more generally. I'll throw in a plug. So as was introduced, so I just started my lab at Baylor this past summer. And we're looking for students, but especially postdocs who are interested in exploring these kinds of questions of both experimental and computational views of RNA biology and how RNA binding proteins regulate their targets. So if anyone's interested, feel free to visit the lab website or reach out, and I'd love to chat more. And with that, I'll stop and thank my lab at Baylor. Obviously, this work was primarily done in Jean Yo's lab at UCSD. Most of the data that I talked about was generated by some great experimental technicians shown on the right here. They did a phenomenal amount of work in doing all of these CLIP-scq experiments. I'll thank our collaborators from ENCODE and CPRIT and NHGRI for funding. And I'd love to answer any questions that you have. And if we run out of time, feel free to email me. And I'd love to chat more.
Thank you, Eric, for an exciting talk. Again, that was a lot of really intriguing data and at the same time, very interesting mechanistic studies. Again, if anyone have questions, feel free to unmute yourself and speak up or type through chat. I will lead with a question from more of an outsider point of view. I don't know RNA biology that well. It seems like from the ECLIP data, there definitely is quite some sequence specificity to where the RNA binding proteins bind. I wonder if it differs. Seems like in some cases you guys have conducted experiment across different cell lines, maybe in different tissue lineage. Does that affect where the RNA binding proteins binds much?
Yeah, so that's a great question. And that's something that we looked at a little bit in that work. So the answer is in the literature, there are examples where you can see differences in binding between cell types for sure. There are cases where, one RNA binding protein's binding site will be blocked by another RNA binding protein that's tissue specific, for example, or, structures can change between cell types and that changes the availability of an RNA binding protein's binding site. From the ENCODE data, we actually didn't see very much of that. So when we looked at what was different between the two cell lines that we had data for, the vast majority of the differences that we saw were just genes that were expressed in one cell type or the other. And so they would be bound in the cell type they were expressed in and not bound in the cell type where they weren't expressed. But we think that has a lot to do with the fact that we mostly, this work was really in two cancer cell lines that are not that distinct. So I think if you go and look at different tissues or different post-mitotic cell types, things like that, then there's more of those kinds of differences that people have seen in the literature.
Okay. I think people are still taking the time to process in their mind all the exciting data. So maybe I'll follow up with another question. Oh, sorry. Go ahead, please. Please go ahead.
Yeah. Yeah, I have a question. Hi. I was wondering whether this project will continue, like now adding some data of structural information, like for example, shape data in this landscape of binding for these RNA binding proteins, because I think the community is quite excited right now by the structural compositions of the RNA, or also whether there is plans of adding long read technology and kind of also developing more in the long read technology. But yeah, I don't know what is the status of the data.
Yeah,
collection.
sure. So yeah, so the project is continuing. I think with COVID, especially, things got delayed, but I believe they're actually trying to release a bunch of data in the next month or so. So we would love to do a bunch of that stuff. What is funded is primarily the same types of data, at least for the ENCODE project, to just do more RNA binding proteins and trying to fill out that landscape. But I know that I think I agree that is very, very interesting. Overlapping, especially like the RNA binding protein targets with what's available from shape and trying to build build that mechanistic understanding is very interesting. I know there are some labs, especially that are experts in the shape technology and stuff like that, that have generated shape maps for the like K562 and HepG2 cells where we have all that data. And so I think there's some very interesting work going on there, but not primarily by the ENCODE group. Having said that, I'm not as involved in the next phase of this, so there may be some efforts to do that, but yeah, that would be very interesting. I think other things like looking more, like you say, at using long read technology to look at some of the correlated splicing changes between more distant events. Obviously, trying to move past just looking at splicing to looking at things like translation rates and stuff like that is also something that I think is very interesting, and we have some interest in trying to go forward with as well. So there's a lot of areas that I think would be really interesting to pursue here that our people in the field are looking at, but that we haven't sort of really started to do.
Thank you.
Okay, and we can take another question. I will ask one short question in the time that people might be coming up with questions. So, in my understanding with the ECLIP assay, you can do one RNA binding protein at a time, and definitely there are probably a lot more RNA binding protein. So how do you think about tackling the rest of them?
That's a good question. It's tricky. So we've been trying to develop some methods to make the method more scalable. especially trying to couple it with tagging approaches. Using antibodies limits the ability to scale to some degree, and there's reasons why for some RBPs you can take some shortcuts and others you can't, depending on how much background there is and how specific the antibodies are and stuff like that. I know there is definitely some method development going on to try to make methods that let you look at multiple RBPs in parallel, trying to map either kind of more similar to like the way DNA's hypersensitivity works for DNA binding proteins, or trying to use other methods to sort of label multiple RNA binding proteins targets in the same experiment. Those are also still very experimentally tricky, but there's definitely a lot of people that are making effort in that area. It just sort of isn't as good yet as sort of going deeply into an individual protein.
I see. Yeah, I think there is a lot of exciting technological investment that is going to be made. Let's give 10 seconds to see if there's a last question.
Yeah, and also throw in, well, if anyone, feel free to interrupt me, but I think the other way that people are trying to solve it for the omics perspective is people are using this data to try to do much better jobs of just being able to predict from sequence where RNA binding proteins will bind. And I've seen a couple of unpublished stories that seem to be very, very good at starting to do that now. So that's sort of the other aspect that is really making progress, is the computational side of being able to do this even without necessarily even doing the experiment.